Hi. Sorry, I'm just setting myself up, ready to go. How are you? How are we this evening? Oh, I am tired. I have been tutoring all day. All day. This is my ninth hour of teaching today. Um, because I've crammed everybody into 2 days. I've got another full day tomorrow, but as always, I'm here to help. So if you have any questions, then please do ask away. My brain is a little bit on slow go today, so I will try my best to give you the part live very late. You should have some rest. Very hard working. Yeah, I know I do need to have some rest. I will definitely be having some rest, but it's half term, which means it's a very different week for me. So the girls were in art class today, and they've had a great day, and I am now doing as I always do, is helping you guys who have your questions. So thank you for the likes and welcome. I am displaying on my other channel. So let me just, I managed to figure out what I was doing wrong. So I think I have now got that sorted. So if you are on YouTube and you are joining me, then welcome. I am answering questions. If you have any, then please do ask away, and I will see what I can do to help you. But yeah. Do we have any questions, any topic areas that we're struggling with tonight that we want to look at? I'm trying to think what I've been doing today. I've had so many different ones. What have we done? We've done the kidneys. We've done patterns of inheritance. Please, could you help summarize genetic modification? So genetic modification is actually a really interesting one, because with genetic modification, what you're looking at is modifying the genetic makeup of an organism. So are we taking a gene from, let's say, a human, the insulin gene, and are we then inserting that into a plasmid? Actually, I did immunology today, B and T cells, with my AQA student. So I will touch on that one in a minute. So let me come back to this genetic modification. So if you've got a gene that is your desired gene, whatever that may be, how are we going to transfer it? Well, the first thing is to think about these keywords, things like the fact that you have got the universal code. So you've got the fact that the code is universal. With it being universal, we can take a gene from one gene. So I talked about having the human insulin gene. So let's say I want, hi Alex, I have a gene and I managed to figure out how to make it not grainy. So I'm hoping this is coming through really well for you on the my YouTube channel. If I've got my gene that's the desirable gene, so this is the gene for, let's say, insulin, what I'd want to do is I would want to transfer that into another organism. Universal, I love it. Universal, degenerate, and non-overlapping. Very good. They are three of the words I'd be looking for when we are talking about genetic engineering. So if I take a plasmid, and that plasmid I know is a circular bit of DNA. Chidima, how are you? It has been such a while. You've been missing. You've definitely disappeared on me for a while, but I hope you are well. So we've got a gene, insulin gene, the desired gene. We've got a plasmid. What we want to do is we want to have it that we're going to cut them and stick them together. So what we need to do is to cut them, we want to use restriction endonucleases. So restriction endonucleases are enzymes that are going to cut at specific recognition sites. So let me just write this down. Restriction endonuclease. So we have enzymes that are going to cut, and where they're going to cut is at recognition sites. So these recognition sites are going to leave us with sticky ends. So what that means is I will have these what's known as sticky ends. So if I've cut the plasmid and I have these sticky ends, what I'm saying is the base that's here whatever the base would be that's at that point there, and that point there, and that point there, they are going to complement the bases that are here. So what would happen is they would want to form hydrogen bonds, but because they are being cut, what we're saying is we are cutting the phosphodiester bond that was between those two strands, and we're using the same restriction endonucleases. So what it then means is if I take this and do it again, but this time I'm going to ungrip them. I'm going to put these two together. I'm going to make these red. There we go. So now we've got red ones, and the red one is part of the human insulin gene. And so what I'm going to do is, by doing this, if I've used the same restriction endonuclease, I'm going to have the same sticky ends, which means that they're going to be able to come together, forming the hydrogen bonds because they complement each other. There's my shapes. Doesn't really work. Let me just quickly correct. Oh no. Let me do that again. Get rid of that. Get rid of that. And so what we get is a new connection there. And so when you have your ligase, what the ligase is going to do is the ligase is the enzyme that's needed to join the fragments together. So my insulin gene, and that's going to be joined together with my plasmid. And because it's going to be joined together with my plasmid, what we're going to end up with is we're going to end up with a bigger plasmid. But this time, that was really not what I wanted to do. Let's try that again. You can tell my brain's not quite as on it as it should be. Is I'm going to have my genes. Let me think, it's got to go that way, which therefore means I need to add this bit in. And there we have, by inserting one of the genes from one organism into another one, I've now created a recombinant plasmid. And that's because it's different from the original. So this is recombinant plasmid. So I've inserted a gene from one organism to another. Hi guys, welcome. Sorry, yes, this is year 13. I'm currently looking at the genetic modification that was asked. So this is for those of you who are OCR A, this is your manipulating genome topic area. So the key thing to know about genetic engineering is those things, as Alex already said, it's universal. It's a genetic code which is universal, so I can take it from one organism and I can stick it into another organism. I can use ligase, which is an enzyme that's going to be able to join the fragments together because of the phosphodiester bonds that are going to be needing to complete the phosphate sugar backbones. I'm going to use restriction endonucleases to cut at specific recognition sites, and by doing that means they're going to have these sticky ends which allow them to come together. And so the other thing to note is your complementary base pairs. When you have your nitrogenous bases, your purines and your pyrimidines, if they have got the same order, the sequence is the same, that complements each other, they will stick together. So that's how we can move the genes from one organism to another. However, we need to make sure that we link in the fact that we need electroporation. We need for the cells that we put them into to actually take them in. So we can either do that by putting a little electric shock in them, and that will make the membranes porous to take in the recombinant plasmid, or it may be that we're going to do a bit of a heat shock to them, where we heat them up really quickly and then we bring them back down really quickly. Hi Christine, I was just revising cellular control and a question came up on the anticodon of a tRNA molecule. I was confused as I thought anticodons, no, okay, so anticodons are located on your tRNA, your codons are on your mRNA. So your codon is the triplet base, which is on your mRNA, which is going to correspond to the code which is on your DNA sense strand. So be careful. Let me add that in, and then I will come back to the bit that's on T cells and B cells. What you need to be aware of when you're looking at this is if I am doing my DNA, so I know I've got my DNA, which is a double helix. I'm going to draw my double helix here. So this is my DNA strand. I am going to copy the code onto the mRNA. So this is my mRNA in red. Let me try making that a small m. mRNA. I need to do this a little bit slower so I'm not rushing. What we have is the antisense strand, which is the template strand on the DNA. We have the sense strand. Now, on my sense strand, if my code, and I'm just making this up by the way, if it were to go A, T, T, C, C, A, G, A, T, C, G, A, T, C, then I know on my antisense strand, it is going to be the complementary. So A pairs with T, T pairs with A, G, G, T, C, T, A, G, C, T, A, G. So that's my complementary antisense strand for my sense strand. So therefore, if I now bring in my mRNA is going to be complementary to the antisense strand, and therefore the A and U. So my mRNA strand should be the same as the sense strand, apart from the position where there is a T, I would find a U on my mRNA strand. So if we're looking at it just very basic, so my DNA strand, if I say ATT is, no, I don't want to do that. Let's try that again. Let's do the other way around. If I do this again, going up. So if I go TAA, then my mRNA strand is going to complement that antisense strand and therefore be AUU. And then what I would do is I would take that and I would then rotate this around just now and rotate it around that way, just to make this make sense, hopefully, even though it's upside down. And then in my tRNA antisense strand, I'm doing this very basic, this would be UAA to match it. So I'd have my DNA, there's my DNA, there is my mRNA, and all I've done is I've rotated it slightly here to make it so you can see, and here is my tRNA. So this is my anticodon, this is my codon, and this is my template strand on my DNA. V, does that answer your question? Hopefully it does. You just finished your neuronal communication. Can I finish module 5 by the start of March? Absolutely. Have you done module 6 at all, Sully? Or are you just doing the revision on your own again? Reminds me. Is it okay to do respiration? As in, when you say, is it okay to do respiration? What do you mean on that one? Do I do GCSE? Yes, I do GCSE, but I haven't actually been doing it because there's so much to do right now. It's exam period. I got confused by the difference between genetic engineering and selective breeding. Selective breeding is where you're selectively choosing organisms that have the traits that you want, and you are then breeding them together. And so because you're breeding them together, you are selecting those traits, you're producing the offspring, you are then breeding the offspring together again. So it's one of those things that You are selectively breeding. Genetic engineering is that you are genetically manipulating the genome and therefore you are affecting that. If you put the work in, you could even exceed that. Yeah, you can absolutely 100%. If you put the work in and you're working at it, you will be able to excel, but it's hard. Right, sorry, I'm just skipping through to see if I've missed any of the questions that you guys have asked. Apologies on if I have. I think we're good on that one. And thank you very much for all the likes, by the way. Wow, 2,000 already. Do I do AQA A-level? Yes, I do AQA A-level as well. I was tutoring tonight. We were actually looking at T cells and B cells tonight. And you have asked me for some exam questions. I will get some exam questions up and going. Oh, look at that. 81 out of 91. Your AQA paper 2 mock. That's amazing. That's so good. Wow. For OCR A A-level, what did you ask? Sorry. Do you do WGEC spec 2? I haven't done that spec. I've looked at the spec, but I haven't actually been teaching that one because it's one of those things. I'm actually getting super busy right now. Everyone's panicking. I get it. Everybody's panicking, and therefore my time is being just reduced on what I can offer. And also I want to work on my study guides, and I can't because I have no time. And I want to work on my course, which I'm going to be offering in Easter and May. And so right now I'm really consumed with building those resources at the moment. So I'm really limited on time. So I'm afraid I'm not. However, there is an overlap in the content. And so what I would say to you is don't get too hung up on the parts of the course that are the same. What I'm trying to say is if it's the same, it's the same. The content's the same. There's just little changes you need to make. So for example, if I'm doing aerobic respiration, because someone said here about the stages of aerobic respiration, if I were doing the link reaction, I was doing AQA, I need to make sure that I'm talking about acetate and not just calling it acetyl. If I'm doing the secondary messenger and cyclic AMP, if I'm doing AQA, I need to be aware that it's adenylate cyclase and not adenylin cyclase. And so it's little things like that that make it slightly different for your exam board, and knowing that. But biology is biology. It's just about where their emphasis is that you need to be aware of. Right. I forgot where I was going with this one. We're looking for some questions, weren't we? I had been working through the which one was I doing? 2023 questions, wasn't I? Oh, actually, do you know what? I'm going to show you. I did a thing earlier, and I was so... It sounds ridiculous. I was like, oh, I really like this resource, and I need to turn it into a study quick recap for respiration. So it's because someone said respiration, and I was doing it with one of my students earlier today. And as I was doing it, so I'm going to put it on here. I love it. Thank you to your cat for watching my stream. Now, this may make no sense whatsoever to any of you, but whilst I did it, it did make sense. So I'm going to put that through. So we were talking about respiration, and we were talking about what happens, and going very back to the basics for respiration. So you should know about glycolysis. Glycolysis, obviously being broken down into pyruvate, very basic. I'm not going into the step-by-step guides. Hold on 1 second. These want to clarify. Does the tRNA and mRNA strands together form the new copied DNA? No. Be very careful. Hold on, guys. Sorry. I will come back to respiration. But these are the sorts of questions that I want you to ask in my sessions, in these sessions, because that's the whole point in it. I'm here to answer the bits that you don't know so that you are able to clarify for it. So if I have got my DNA, I'll do this again, my DNA, which was TAA, and I had my mRNA, which is AUU because it's ribose, so there's uracil, not thymine. When I leave the nucleus, so I'm moving through the nuclear pores, the mRNA is the only bit that can get through the nuclear pores. When the mRNA joins to the ribosome, what you have to remember is that your tRNA actually has got a very specific shape to it. So the tRNA has a specific shape, and if you are a AQA student, you have to know that in more detail, and the fact about the clover leaf, although I actually do give it to you even if you're not AQA, if you're an OCR, it's still important to know. The tRNA molecule, so this is my tRNA molecule, is a polynucleotide. It's a polynucleotide that has a very specific shape, and there's only one region of it, which is the anticodon. Okay, and so therefore what you should know, the reason it's got its shape is because it will actually form hydrogen bonds within its structure, which gives it the recognizable clover leaf shape that we're used to. So when the mRNA comes down to the ribosome and attaches to the ribosome, what you have is the tRNA molecule will actually make its way, if I can get this to work properly, but it's not going to play ball for me. Give me a second. Move this down here one second, and I want to just delete some little bits of it. So this is my tRNA molecule. I'm just going to get rid of the notes down there. And the tRNA molecule has got, let's say, a 1 amino acid here, and then we're going to go, we've got a different amino acid now. We're going to give it a triangle this time. All about shapes. And so what you're going to have is the tRNA molecule will have a very specific anticodon which corresponds to the correct amino acid. And so when they are joined together, that can move around. So when it is going to complement, it's going to come down and complement the mRNA if it has complementary base pairing. And that will therefore mean that this amino acid is going to be the first amino acid. Then we have the next one with my triangle, and that's going to go, and it's going to come into this position here. Okay, now obviously I'm not to scale with my diagrams because I'm just drawing this as quick as I can, but what you will know, or what you should hopefully know, is when I have a amino acid, my amino acid obviously has got its amine group, it's got its R group, it's got its carboxylic acid group, and if I draw another amino acid, doop de doop de doop de doop de doo 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 doo. Does anyone else do this? I do this all the time where I'm like, this is where I sing myself to sleep. Or I'm not going to sleep. I'm literally just trying to keep my brain active. Here I have two amino acids coming together. And so therefore what I'm going to do is I am going to have a condensation reaction occurring because I'm going to remove the water from here. And then what's going to happen is I'm going to join these two together through the formation of a peptide bond. And the key thing to note is your ribosome is a molecule. It's an organelle that's made up of ribosomal RNA and protein, and it acts like an enzyme because it catalyzes the reaction to join these two together through that peptide bond. Oh, I finished The Night Manager. I'm so disappointed. I'm so disappointed I don't get to watch it again. But there's a season 3 coming. So if you're only watching season 1, season 1's good, season 2 is good, but season 3, hurry up. I love it. I do. It's a good one to watch if you like that kind of thing, which I do. I haven't actually watched any TV this week. I've been ill again. It's like a second cold that's come through. You might need to start reading ahead. Oh, who is needing to read ahead? Everybody should be reading ahead. Your teacher's still on excretion. The thing to remember about your teachers is your teachers are teaching them in whichever order that makes sense to them. And so therefore, I would say don't panic too much. It's about making sure that you are understanding it. As a teacher, we can fly through the content too quickly, and that can result in you not understanding it, or we can go too slow to get you to understand it and we don't manage to finish the content enough. It's kind of why I made my YouTube channel for you, because that's got everything you need on it so that you can actually go and watch. You can look ahead, you can catch up, you can recap. It really depends where you want to go, but I wanted to make sure that there was the content there for you, because even I know when I'm teaching the content, it's a struggle. I can do it. I've done it. I mean, I've taught the whole A level in a year before. I've taught AS in 5 months. I've done it in very accelerated time frames, but it depends who you are as a student, if you're able to take on that information at that faster pace. And if you're not, then you need to be recapping it again, and that's challenging. But yeah, just to show you what I did with my student earlier, I was saying about the respiration topic. So I put this through. You've got glucose going to pyruvate. Pyruvate can go down the anaerobic respiration, and it's all about regenerating that NAD. Whether we're talking about it in the ethanol fermentation in yeast and plants, we've got our pyruvate, decarboxylation, ethanol. Ethanol becomes ethanol if we have, obviously, the same thing happening up here. My reduced NAD is coming in, and my NAD plus, my oxidized NAD, is coming out. So that's if it's anaerobic respiration. And then I said, okay, well, if it's not anaerobic respiration and it is aerobic respiration, what happens then? The pyruvate needs to get into the mitochondrial matrix. So the pyruvate has to get through the outer membrane and the inner membrane to get into the matrix where Link's reaction and the Krebs cycle is going to happen. Now, if we've got the Link reaction, and we were talking about how pyruvate gets converted into acetyl coenzyme A, and as I said to you, if you're AQA, that should be acetate coenzyme A. And then the acetyl coenzyme A is going to come into the Krebs cycle. And so the blue is showing you the Link reaction, that pyruvate is the substrate, acetyl coenzyme A is the product. Acetyl coenzyme A has to come into the Krebs cycle. And so when you look at the Krebs cycle, excuse me, what you've got is the oxaloacetate gets converted into citrate by the acetyl being added to it. So the 2-carbon molecule getting added to make a 6-carbon molecule. And so the coenzyme A is a coenzyme. It's an acceptor of hydrogens. It picks them up and it drops them off. In this case, when we're talking about the coenzyme A, it's picking up the acetyl group and it's dropping it off at the Krebs cycle. And then the Krebs cycle is obviously going to go on in the fact that it's a cycle. It's going around. And so the citrate gets decarboxylated. You've got your NAD coming in, accepting hydrogens. The 5-carbon is going to be decarboxylated. Now, there's loads of intermediates. And I spoke about this with my student earlier today. And we talked about the fact that I don't show it, but I've got a 4-carbon, I've got a different 4-carbon, I've got a different 4-carbon, I've got a different 4-carbon. We don't talk about those intermediates. What we say is we go from oxaloacetate to citrate to a 5-carbon back to a 4-carbon, and all these things are happening. So the key thing for you to know is knowing, what do I actually need to know and memorize for the exam. So things like oxaloacetate and citrate, you have to know for OCR A. Acetyl coenzyme A, you have to know. You can't skimp those ones. You can't just go, oh, it's a 4-carbon, it becomes a 6-carbon. That's not good enough. You need to know that content. And then we talked about the fact that, well, when we get on to oxidative phosphorylation, what's going to happen is understanding where the electron transport chain and ATP synthase is. So the inner mitochondrial matrix, no, try that again. The inner mitochondrial membrane is this section here. The orange bit is the matrix. And so I was trying to show them the difference between the outer membrane and the cytoplasm and the inner membrane, which could be the cristae, the folds of the inner membrane, or could just be the inner membrane. So when we were looking at it, I was saying, well, here's my mitochondria, mitochondrion, singular, and the yellow is representing the cytoplasm. So up here, we're looking at this section where I have the outer membrane, the intermembrane space, the ETC, and then I've got the matrix. I've got matrix on both sides. So this is the cristae, the fold of the inner membrane. So I quite like that diagram and went through the whole what happens, etc. With regards to respiration in our session today. But I was like, ooh, I really like the way that that was represented. And then we went on to say, okay, so what's the point in glycolysis? 2 reduced NADs. What's the point in link reaction? 2 reduced NADs. What's the point in the Krebs cycle? 6 reduced NADs, 2 reduced FADs. All of these going off to the oxidative phosphorylation as part of the inner membrane. And that then leads to, well, if we've got water being produced as a waste product, because it's oxidative phosphorylation, we have oxidation and reduction reactions going on in the fact that the oxygen is the last electron acceptor. That is going to accept the electrons and accept the hydrogen ions, and therefore becoming water. And what do we get? We get ATP molecules. So this was like a skimmed way of me showing you the work that we did this afternoon, me and my student, but I quite like the diagrams, so I thought I'd share them with you here, because actually, why not share if I come up with these things and I do it on a quick basis with you guys now. Obviously with my clients that pay for their sessions, we go through and go into it in much more detail, and I make sure they actually can say it back to me rather than me just saying it at you. And that's the thing that I would say, YouTube is a wonderful thing. Having my videos, having the videos to watch is a really good thing. Absolutely 100%. However, how are you assessing that you're actually understanding the content that you're learning? How are you assessing your knowledge that I am giving to you? Because yes, I know it, but I'm not the one sitting the exam. You're the one who's sitting the exam. You're the one who has to get this down on the paper to prove to the examiner that you know what you're talking about. And so that's where students fall down, because they're like watching it going, yeah, yeah, I get it 100%, I know that. And then I'll ask them the same a question, which is, I don't know, why, if there's no oxygen available, does aerobic respiration not occur? And they can't then relate that to the fact that, well, if there's no oxygen to be the last electron acceptor, the electron transport chain can't pass on the electrons, which means the coenzymes can't be oxidized. If the coenzymes can't be oxidized, they can't go back to pick up more hydrogens, and therefore the whole process of the linked reaction Krebs cycle stops, because there's not enough coenzymes for picking up the hydrogens, accepting the hydrogens, and therefore changing the pyruvate into acetyl groups, or the citrate to get back to that 5 carbon, etc. So if you can explain that, if you don't understand those parts, then you're not going to be able to answer the questions in the exam. And that's what they're assessing you, those application skills. It's about you understanding not just what you've memorized on the topic areas, it's how it all interlinks. Is further maths tough? I would say absolutely 100%, because there's no way I'd be doing it. Maths is hard. Is further maths hard? It depends on whether you are someone who likes maths. Could you possibly explain the zero dilutions with the agar plates? their calculations are a bit confusing, of course. What time are we at? 31. Let's go on to the parts to do with zero dilutions, etc., and with our plates. So let's say I have a stock solution, okay? I've got a stock solution, and in that stock solution, I have got, I don't know, how many bacterial colonies, and therefore bacterial cells, have I got in this stock solution? I don't know. What I do know is I am going to do a zero dilution. I'm going to do 1, 2, 3, 4 zero dilutions. 1, 2, 3, 1, 2, 3, 1, 2, 3. Okay, so I'm going to do a 10-fold zero dilution. So what that means is I am going to be putting in 9 centimeters cubed of distilled water into each of mine. 9 centimeters cubed of water is going to go into my test tube. Let's call this A, let's call this B, let's call this C, let's call this D. And what I'm going to do is I'm going to place 1 centimeters cubed of my stock solution into A. I'm going to put 1 centimeters cubed of A into B. I'm going to put 1 centimeters cubed of B into C. I'm going to put 1 centimeters cubed of C into D. Okay, so what have I got? In A, B, C, and D, I've put 1 centimeter cubed and 9 centimeters cubed. So I have a total of 10 centimeters cubed. That's my volume inside A. That's my volume inside B. That's my volume inside C. That's my volume inside D. Each of them have got 10 centimeters cubed. So I'm going to do a volume cell table. And I'm going to put here, I've got my stock, I've got my A, B, I've not got enough room, C, D. Okay. So, how would I work out what I've done? Well, my volume, let's say my volume in my stock solution is 50 centimeters cubed. So I have got 50 centimeters cubed inside my stock solution, but I don't know how many cells I have in there. What have I done? Well, I have taken 1 centimeters cubed out of the 50, and I have added 9 centimeters cubed to equal my total, which is 10 centimeters cubed. So my volume is 10 centimeters cubed. So how have I got 1 centimeter cubed out of the 50? That would be where I have divided 50 by 50 to give me 1 centimeter cubed. Just bear with me when I do this, okay? So I've divided 50 by 50 to give me 1, and now I have got 1 centimeter cubed in 10 centimeters cubed. Now I'm going to take 1 centimeter cubed of this to go into B. So I have divided 10 by 10 to get to 1. Therefore, I'm going to have diluted it down by a factor of 10. Again, I'm adding in 9 centimeters cubed to give me 10 centimeters cubed. Search up in Joshua AQA. Give me a second. I will look that one up in a second. Let me just finish my made up question. Plus 9 centimeters cubed, 10 centimeters cubed, and 1 centimeter cubed plus 9 centimeters cubed equals 10. Is everyone following me up to this point? So I don't know the cells yet. All I know is about the zero dilutions. How many times have I diluted it? Now, let's say I take out each of A, B, C, D. So A, B, C, and D. And I am going to remove out of that 0.01 centimeters cubed. And I'm going to spread 0.01 centimeters cubed out of each of the test tubes. Okay. And I am going to spread it and see what happens. So I might get this. I might get that. I might get this. And I might get that. Let's do a bit more on this one. Okay, so this is the size. This is what's grown. This is the colonies. Now, if I'm looking at this and I'm working my way backwards, so I have got my cells, I've got my volume. I'm going to start with this 1D. So I'm going to start at the bottom. In my plate, I have got, try that again. In my plate, I have got 3 cells and the volume was 0.01 centimeters cubed because that's how much I took out of my sample. Okay. So now what I want to do is I want to put that back up into the test tube of D. So to get it back, I had 10 centimeters cubed in my test tube. So how do I get back to 10 centimeters cubed? I had to divide it by 100. If I'm going back by timesing it by 100. Nope, by 1,000. Hold on. 1, 2, 3. By 1,000, not 100. See? There's me doing it, having to think about it. So I would times it by 1,000. So I would therefore times this by 1,000, which would give me 3,000. What am I doing? If I'm going back to C, I'm timesing it by 10. Because I'm timesing it by 10 to get back up to C. Because I took 1 centimeter cubed to go down, I divided it by 10 to get my 1 centimeter cubed. If I'm going back up, I would times it by 10. So therefore I need to times this by 10. So then I get 30,000. Going back up to B, I'm going to times that by 10. Again, times that by 10. So therefore I get 300,000. And if I'm going up to A, I'm going to times it by 10 again to get back up there. So, and now to get up to my stock solution, I'm going to have to times it this time by 50. And so therefore I would times that number by 50 to work out how many were in the original stock solution. Let's see if I can get the calculator on here and work this out. So I had, what did I have? 3 million. I would have ended up with, in my stock solution, 150 million. So one of the things you can do is count by the factor of how many am I working my way back. And so this is what I would do. So I've got 1, 2, 3, 4, 5, 6. So if I did power of 6 times 50. Yeah. So I would have, I could have done 3 times 10 to the 6 because I was going back factor of 10 to the power of 6 and then times that by the 50 to get my 1,500, 000,000. My brain doesn't work in maths in the same way as other people's brains do. And so therefore, sometimes I have to visualize it in a table. And I found that table method works for me. I used to do it with the going back and using my little diagram this way. And then I would get myself a bit confused. And so when I started doing it with this little table, it just seemed to work. It made sense for me. I was able to see what's the volume, What did I do going down? So how could I go back up? Now, the reason I want to show you this question, and I haven't forgot about you saying the 2018, I will have a look for it in a minute. If we looked at C this time instead, now remember I've still got 0.01 centimeters cubed coming out. So I'm going to do the same again, volume, cells, and then I've got stock, A, B, C. So I've still got the plate, and the plate has, excuse me, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So my plate has got 11. I would be doing exactly the same thing. I had 0.01 centimeters cubed. So to get back up here, I need to times 10 to the power of 3. So therefore I need to do 10 to the power of 3, then 10 to the power of 1, times 10 to the power of 1, and then times the 50. So if I do it this way, la 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 la. Unit 5, right, give me a second. So I did 11 times 10 to the power of 3, 4, 5. Of course it's 5 because I've got one less. Oh, no, 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 don't do that. Times 10 to the power of, go away. 11 times 10. Oh, I'm doing it all wrong tonight. 10 to the power of 5 times 50 would therefore give me my total stock, which cells wise would have been 50,000, 55 million even. And so therefore, which one of these is a better way of estimating the population size within my stock solution? Actually C's better at using, because it gives me a number that is closer to the number that would be in the stock solution. By using D, by using a dilution that's gone too far, I have too few cells growing. That would give me a much larger estimation. And so therefore it's about factoring all these different things in. And that's the kind of question that has come up in the past. Right, let me see what we're looking for. I'm looking for AQA International, Unit 5, 2018. Let's see if I can quickly find this one that you're mentioning. International Biology 2018. Let's look. Do 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 do. 2018. Nope, it's not there. Do you mean AQA? International AQA in 2018? Let me see. Let me see. Let me see. The 2019 one I found. I just did a paper 1 OCR A, got 53, 56, 3 marks off an A in 20. That's great. That's a really good grade. Well done. Any tips to improve that now? Yes. So what you need to do is you need to really go through that paper, find out where you missed the marks. What was the reason you're not getting the marks? Is it your detail? Is it your understanding? Is it reading questions? What is it that's holding you back? Is it that you didn't know the content? And so if you can work your way through that paper, not memorizing the mark scheme, because that's a waste of your time. They're never going to ask those questions again. What's important is, well, this is a perfect paper. Sorry, I've just downloaded a different paper and I'm like, oh, this is a really good one for muscle contractions. I might just download that and save that. But that would be my advice, to go through and find out where you lost the marks, what caused you to lose the marks, and that will help guide you. Some reasons for topic knowledge and a lot of 6 markers. The 6 markers, the key thing that comes through for the 6 markers, I'm sorry, some of you have been writing loads of questions in here and I haven't seen it. A lot of problems with the 6 markers is that students don't read the question properly. What they do is they half read the questions. Can you send me a copy of one of the international AQA papers that you've been using so I can look at what you're referring to? Because I use the CIE ones. They're really good. But I haven't actually looked up at the AQA. I didn't know AQA did international papers. This is the thing, there's so many different papers out there. I've got all the AQA actual papers, but if you're saying the international AQA, so do we have international papers? No. It's not letting you copy and paste it. I love it. So can you send it to me in the email? Send it to ukbiotutor.com and I'll have a look there. There's loads of questions though out there that you can use for serodeletions. Thank you. So yeah, it really just depends. Figuring out, going back to how do you improve, figuring out what works for you doesn't necessarily mean it will work for somebody else. Just to be clear, okay? Just because something works for someone else and they try and teach it to you, you might not get it. So if I give you example, Alan's always trying to, I always ask him questions. He's always trying to help me understand things. And whenever we talk about a content that I'm not getting and he's trying to explain it to me, some things for him just make sense. He gets it and he's able to then use that in whatever he's doing. Now he will try and teach it to me and I'm like, I'm not getting it. I really don't understand it. Even though he's telling it to me, he's speaking the language. It's like when I'm teaching, he's speaking the language that I understand and yet it doesn't sound like a language I understand. I try so hard, but it's just not computing in my brain. I don't understand what he's saying because it's not the way that I think. It's not the way that it works for me. And so therefore I've taken a long time to figure out what works for me. So the one thing I would say to you is looking at your own answers. Oh, perfect. Thank you. Let me check if it's come through to that one. It's not come through yet, but hopefully it will come through. Yeah, I've taken a long time to figure out how my brain works. And so I know that my little tricks work for me, but that doesn't necessarily mean they'll work for somebody else. Oh, let's look. And then it was the 2018 one you said, right? How interesting. That was the multiple choice one I was just talking, the muscle one I was just talking about, but that was shown for 2019. How interesting. Coleoptiles. Nice. Which statistical test would you use? Have a look. Which question are we going to? Diluted blood. Ooh. So one of the things I would say is a little bit of a caveat using these papers. These papers, I can see already, these are good questions, sets of questions. However, be careful you don't go into learning too much detail, although you're AQA, so actually you do need to know about how to use the hemocytometer. I can't even say the word on that one. Cover slide, but they do look like they're good questions, to be honest. Oh, here we go. No, that's not the one they wanted. Oh, these are really good papers. Oh gosh, actually maybe not. Some of the I'm now looking, I'm like, yep, that's definitely international. Yeah, exactly. This is the thing. It's once you've got good sets of questions, it is a positive, and because you're AQA, it does follow the rule of what you guys are doing. Have I missed the question though? I can't see. I thought I saw it. Was it the one about the different blood concentrations, sodium chlorides? But that's not. I automatically thought that, but I have a look. It's less about the hours and more about the quality. Yeah, it's definitely about the quality of work that you are doing, not the hours that you're doing it now. It's really hard to know what's right for you. Do you think that I can make a comeback from a D in bio at A-level? Absolutely. It depends what's holding you back from getting the grade. So I've had students who have gone from, where are we now? Let me think this through. We are in February right now. So I've had students go from E's to A's in the space of the time period. So the student that I worked with went from an E to an A in the space of 4 months, and it was all down to practice. It was down to them doing, so they were one of my trial students on AIQ Chat when we first launched it. They were the school that I worked in, who I built the students. I built AIQ Chat for these students, and so they were part of the trial pilot skill, and they were then the first customers of AIQ Chat, and they used it religiously every day in the right way though. So I don't talk enough about AIQ Chat, and I know I should promote it, but I kind of get distracted and I just end up talking about myself. But they used AIQ Chat every single day, and how to use AIQ Chat is you've got to use it by doing the practice questions, and when you get it wrong, chatting back and forth with the artificial intelligence tutor until it is able to get you to understand the answer. And so they did this every day. They did one assignment every day, which was 10 multiple choice questions. Just before a test, they would do more, but they would only do those questions every single day up until their actual exam, and they went from an E to an A grade. Now this was someone who had English as a additional language, so it wasn't their first language. This was someone who just needed to push through the hindrance of not wanting to put things down on paper. And so the more that they practiced, the more that they worked through their understanding with the AI tutor, the more they were able to formulate their answers to think like an examiner. And when it came to the actual exam, they were able to put across what they actually had learned, and it was amazing to see. So yeah, 100% you can get there, but it's about doing it the right way. It's called aiqchat.com. It's a website me and my partner built for students, for OCRA students who were studying basically I wanted to give it to my students as a homework. And so I did. They would do it as a homework every day. And then when I removed it from them, they were like, give it back to me. And so they became my first paying customers. And that was way back. When did we first start that? 2023? Um, the IA question, hold on, I think I might have that one. Give me a second because I've got the actual, it was an AQA one you were talking about, right? I did one the other day there with my group tuition sessions because I have an AQA student in my group sessions. And so whenever we're doing something which is relevant to both exam boards, I bring in the AQA questions as well. Um, let me look, let me look, let me look. So hold on, that was OCRs. Here we go. I think I found it. Was it this one by, oh no, that's not what I wanted to do. I hit the wrong button there. Was it this one by any chance? So it was talking about a student prepared a range of oxen concentrations using zero dilutions. The original oxen concentration had a concentration of 1.0 times 10 to the 3. Was it that one? Talking about elongation of oat coleoptiles. Actually, I think this is one that I made up. This isn't it. This is the one that I've made up for my study guide. I was like, no, I recognize that. It's because it's mine. So yeah. Oh, my live's gonna end soon. I've not been going for an hour, have I? What time is it? Oh my gosh, I have. I've been going for an hour. There's a 6 marker about preparing a set of IAA from 10 to 3 to 10 to 2. Oh, okay. Yeah, I've just realised this isn't it, because this is one that I've made up. AQA, hold on, let me see. Past papers. IAA. I'll find it for next time. Yeah, definitely find it for next time. And it was definitely in an AQA paper, right? Would that be paper 2? No, it's not paper 2. Hold on. Ah, right, okay, I'll need to have a look. Can I ask any other papers or questions you recommend doing after I finish the OCR A past papers? I would say if you're struggling with application questions, but be very careful using other exam boards that you only do questions that are relevant to your course. I would say EdXLB is really good for the application side of questions. I've found their questions when it comes to the graphs, when it comes to evaluate things like that, those questions are really good. So I would suggest they are a good shout to use. CIE, I International A level. I also like those ones. But again, it's down to knowing what to use and how that relates to your specification, because when you start going off topic areas, when you start learning things that are irrelevant to your exam board, you will lose marks potentially if you're answering questions with answers that are not relevant. If you're AQA, that's different because you've got the 25 marker, so you can add in those extra bits. But for OCR, there's so much content you have to learn that they don't want you to learn anything more. They just want you to learn that, and then they want you to apply that to the situation. So the more you get used to the OCR ones, and I would say if you haven't done the AS papers, I definitely suggest doing the AS papers because that will really confirm your knowledge of the module 2, 3, 4 content. And therefore you can also see, like, I can predict whenever I look at the year 12 questions, I can predict where they would go if they're going to turn this into a year 13 question. So using them and then thinking to yourself, what could the question be? Where could this go? Oh, okay, so you've done all the AS papers already. Perfect. Then I would suggest basically Edexcel B would be one I'd suggest, and also AQA is good actually. Some questions in there are quite good. The comprehension part of paper 2 is good for being able to read the long stem questions, but be aware they do have some differences. Like whenever they do graphs, they always talk about 2 standard deviations rather than it being 1, which OCR do. So little things like that do make a big difference. But guys, I am going to finish now. I appreciate you all joining me for tonight and asking your questions, because this is what I wanted this to be about. You asking questions and me being able to help those of you who maybe tutoring isn't for you. And so I do want to always offer this on a Wednesday. So this is the one time I will be available. The thing is, say I do bad on a topic on PMT, the topic questions are from past papers, so I know the mark scheme sometimes. Yeah, so that's... So if you are doing, to answer your question, if you're doing your topic questions, now just be aware the topic questions on PMT, and I'm not putting PMT down because actually I do think it's a really good website and I do send my students there. But those questions are only up to 2022. They're not the most recent ones. So make sure you are doing the more recent papers too. If you do bad on a topic in the topic questions, then don't memorize the mark scheme. Figure out why you didn't know the answer to the question. Figure out what's holding you back and go and do the revision of that topic area, because if you're getting the answers wrong, you don't know the topic. And if they were to ask you that topic area again, they're not going to ask it in exactly the same way. So memorizing that mark scheme is not going to help you. What's going to help you is knowing the topic area, knowing why that was the right answer. What was it they were assessing you on with that question, for example. But I am going to go now, guys. So thank you for joining me, and I will be back again next week on Wednesday. So please do come join me again, and keep watching my videos, keep liking, keep commenting, keep sharing, because the more you do that, the more I can earn a little bit from my YouTube channel, which is great. It means I can still keep doing this, and this is what I want to do. I want to help you guys as much as I can, but obviously I have to earn a living, and this is my 9th hour of teaching. So I am going to say good night, and I will see you all next week.